adult. So this is a tube that is just representing an adult intestine. He isolated from this intestine the epithelial cells, which are the cells that are lining the intestine. And he took the nucleus from these cells and he injected them into eggs which had the nucleus removed. And this then gave rise to an adult frog. So this was the first animal that had been cloned using DNA from a mature cell. Now, sometimes the experiment didn't work quite right and he got sort of partial embryos, but we're not going to talk about that today. But the fact is that with a relatively good success rate, he was able to get adult frogs. So this was absolutely groundbreaking because it proved that the DNA in an adult mature cell was not altered in a way that was irreversible. And that that DNA still had all the potential, because once it got into this oocyte, the genes in that egg now could no longer be intestinal genes, but they changed their entire gene program to be embryonic and to form a live adult frog. Now, this process of cloning, so this is cloning a frog from a single nucleus, has been done now in quite a number of organisms. You guys some of you may remember when Dolly the sheep was created. This was Ian Wilmot in '97. And what Dolly was created, essentially the same technique. A nucleus was taken from an adult cell, in this case it was an udder cell, and that was injected into an enucleated egg cell. What made Dolly unique is she was the first mammal to be cloned. We keep going up in the kind of complexity. Once you clone a mammal, what is the next question? So we'll get to humans in a minute. But Dolly made a big splash because of this. Now, I have